In this journey, we are focused on some issues, among the most fascinating and mysterious emerging from the current knowledge on the origin and the evolution of life. This journey includes the transition from non-living to living organisms in the primordial Earth, as well as a subsequent evolution from the first cell to the present biological world, hence the title Life in Progress. So, to begin with, what do we know today about the formation process of the first living beings and what they looked like? Well, what I may state first is that in the last few decades there was a remarkable progress in knowledge. Yet, from a certain point of view, this has paradoxically raised more questions rather than providing answers. And this is one of the most fascinating aspects of the whole story. There are still many aspects to be understood, which deserve in-depth analysis. To start telling the story, also in chronological terms, I may say that the first experiment that in some sense aimed to explain how life appeared on the planet is the one everybody knows about. It's the 1953 experiment by Stanley Miller, who tried to reproduce the conditions he believed could be found in the primitive Earth in a system reconstructed in a lab. A confined system containing all the components that he believed were present, the atmosphere at the time, ammonia, methane, hydrogen, and of course water vapor. He also included a source of energy in the form of electric discharges, an obligatory prerequisite for chemical synthesis to take place. In this way, he managed to demonstrate that under such conditions a wide range of compounds arose, which are fundamental building blocks of all living organisms. For instance, amino acids, the building blocks of proteins, metabolism intermediates, even our own metabolism, such as lactic acid. In other experiments subsequently carried out by other researchers by following similar procedures, nucleotides, the basic components of nucleic acids, were also found. An even more fascinating aspect is that later on these compounds were shown to be pervasive. They permeate interstellar spaces, as shown by spectroscopic analysis. Thus, the first lesson is that there is a chemistry of life that forms spontaneously and is probably widespread in the cosmos. This indicates that the generation, not for sure of life, but at least life's fundamental building blocks, seems to be a simple process. Even more fascinating results were obtained by subsequent investigations. Since the 80s, for instance, some things were started, things that are still underway. Such investigations have been focusing on hydrothermal vents located on the seabed. High-temperature gases come out of these cracks on the Earth's crust, gases rich in vapor types of organic and inorganic compounds. Some simulations and experiments made in the lab suggest that these formations may give rise to new and interesting mixes of compounds relevant to the chemistry of living beings, thus foreshadowing real metabolic transformation, something much more complex than Stanley Miller's chemistry. Certainly, we are not talking about real life. However, on the basis of such discoveries, all today's scientists tend to believe that life appeared inside these formations, where a chemical complexification on the path to the formation of primordial organisms may have taken place. What does all teach us? Well, with no prejudice the fact that nobody has ever succeeded in reconstructing life in a test tube, this discovery suggests that a suitable, all but rare combination on the Earth of physical conditions are available. So again, nobody pretends to reconstruct life in a test tube, but we know that there were physical conditions that made it possible for organic and inorganic compounds to generate a chemical evolution. So, as you have rightly stated, though nobody has succeeded in reproducing life in a test tube, was it possible to have an identikit of the first cell? In a sense, yes, at least as a first approximation, by extrapolating from the molecular design of the organisms that today live on Earth. However, before delving into this topic and to clarify it better, I think it is appropriate to outline that chronological development of the various eras that have marked the geological, chemical, and physical evolution of the Earth, just because life made its appearance in this scenario. As you can see in the scheme being showed, the Earth is around 4.5 billion years old. This is a very precise dating, and based on the available evidence, the first forms of life, at least those documented, date back to 3.8 billion years ago. They probably existed even earlier than that. Of course, when a certain finding can be dated, it generally means that such a specific form existed sometime earlier. To clarify the scenario, however, we need to add that between the 4.5 billion years of the Earth's formation and the 3.8 billion years, regarded as the age of the most ancient form of life, there is a time lapse in which the Earth was completely uninhabitable. As everyone knows, the Earth was initially covered in lava with extremely high temperatures. It was bombed by meteorites and water was missing. Many hundreds of millions of years had to elapse before the Earth became fit for living, although the timing of such transition is not clearly understood. Such a remark still tells us something important, namely that life appeared on the Earth in a very short time. And it appeared in a very short time as soon as the chemical and physical conditions made it possible. For life to exist, we need a temperature that is not too high. We also need water, all elements that appeared far later than the period in which our planet was formed. What we may say, in some sense, is that the appearance of life, and I like to use this definition, was a rolling start. Quite likely, organisms were formed as soon as the conditions allowed it. As for this primordial organism, quite obviously, we cannot have a real picture of it. Yet, it was plausibly reconstructed based on a very simple criterion. Its molecular features are those which are shared by all the currently existing organisms. If such features are today generally retained, they must also have existed in the Ancestor. This latter is called LUCA, an acronym that stands for Last Universal Common Ancestor. We actually do not know whether other primordial organisms existed at the same time and then became extinct. We will never know because they left no traces. However, based on the aforementioned criterion, we can draw an approximate identikit of the ancestor. And based on this very same criterion, we also know that we all descend from this only primordial organism, which quite likely arose at these hydrothermal springs. So, we are all related. All living organisms are related to one another, and we too. We are all descendants of this small, simple cell. Simple, yet not very simple, because we can infer that concerning the molecular complexity, the LUCA was not so different from today's organisms. For instance, these LUCA, last universal common ancestor, had proteins, nucleic acids as today's organisms. It had a system of protein synthesis, of DNA duplication, of cell duplication, a genetic code, at least very similar to the ones of existent organisms. This is an extraordinarily interesting observation as far as the understanding of appearance of life is concerned, because it tells us that in terms of complexity there is an abyss between our earliest, most elementary ancestors, at least those we know of, and the mix of biological compounds that preceded them and that may have formed spontaneously. So the simplest form of life is something that certainly was very far from what we know today. At this point of the journey, a question comes to my mind. What happened and why in the subsequent developments of this common ancestor? Well, this is evidently a very complex question in that it virtually includes the whole history of life from the last universal common ancestor to our present days. Therefore, it must be tackled step by step, focusing on some elements among all the possible ones. In terms of methodological approaches, those who worked on the issue focused on two main aspects: trying to reconstruct the morphological features of the forms of life that have been appearing in the course of Earth's history, so from the last universal common ancestor onwards, analyzing the molecular features of the existing organisms, which can tell us a lot about the evolutionary process driving the formation of new organisms from the pre-existing ones. Besides this, the molecular analysis has been clearly demonstrated that the tree of evolution has only one organism at its roots, the LUCA, as we have already mentioned. Well, to start with, we know that soon after the first microorganisms appeared on the Earth, 3.5 billion years ago, fossilized heaps of organisms were formed and pervaded the whole planet. These are called stromatolites. Stromatolites are microscopically detectable. As you can see then in the video, and those you see here were filmed in Australia, but stromatolites may be found in many other areas of the world. And these are only the stromatolites that have been preserved up to now. We're talking about huge heaps that have fossilized. They are probably photosynthetic bacteria, so in a relatively short period of time, life succeeds in colonizing the whole planet in such a massive way as to leave a geologically detectable trace. Therefore, life is a rolling start in this sense too, because it becomes quite rapidly a macroscopic feature on the planet. This is not surprising though, as microorganisms reproduce fast. So the fact that in just 100 million years they colonized the whole planet is a scenario that fits perfectly the expectations we may have in this respect. However, after this initial acceleration, we observe a stasis that lasted no less than 2 billion years. After the first microorganisms appeared, nothing else occurred on the planet. Life continued to be there, widely spread, yet in the same forms that had appeared 2 billion years earlier, with no change taking place. I like to use this image. If a witness would have observed the beginning of life, and if a witness would have watched its developments, while considering the almost complete stagnation of the subsequent 2 billion years, well, then this witness may have expected that nothing new would happen and would ever happen over these 2 billion years. So it is perfectly reasonable to wonder why, after this tumultuous start, a period of stasis followed. And of course, this leads to another question. What is the driving force of evolution? Obviously, we now need to talk about the dynamics of evolution to set the proper framework. And if we talk about evolutionary dynamics, we cannot avoid mentioning Darwin. The theory that Darwin developed certainly changed our views regarding the mechanisms of evolution. Very briefly, Darwin's theory postulates that the casual generation of variability is at the core of evolution. In species, for each character, there are several variants, and these are generated randomly. No variant is ex ante more or less suitable to guarantee the survival of the individual. Only ex post variants do the selection, in a sense, prefer those variants that are most suitable to guarantee the survival of the species. In short, this is what Darwin's theory states, and to explain it in practice, we may introduce a paradigmatic example that is also mentioned in textbooks. And this example regards this moth, a moth whose scientific name is Biston betularia, that lived and still lives in some areas of England. This small butterfly alights on the trunks of several trees, preferably birch trees, as their trunks are light-colored. These butterflies are also light-colored, and in the 19th century, after the Industrial Revolution resulted in the release in the atmosphere of huge amounts of soot, they became darker following tree cortex darkening. Clearly, the light color was a mimetic defense against the attack of predators, so their changing color from light to dark was simply an adaptation that enabled these butterflies to camouflage in this new context, so as to protect against predation. This example shows an evolutionary change consisting in the random appearance of a character's new variant. And the mutation that made these butterflies darker was also identified. And it was dated back to more or less the beginning of the Industrial Revolution in the early 19th century. Thus, according to the Darwinian view, which certainly explains a lot about the evolution of the species, well, this is driven by the selection of so-called random generated variations, which in turn is promoted by changes in the environmental conditions. If we go back to the story we began with, it is immediately evident that besides the selection of the fittest, other factors exist. I would say many other factors playing a substantial role in the evolutionary process. And now we are going to document such aspects. Let us return to the story we were telling earlier. 2 billion years of stasis followed the appearance of the last universal common ancestor. During this period, the only existing forms of life were of coarse, unicellular, and primitive. In technical terms, we talk about prokaryotic cells. These enclose in their cell membrane all the components scattered randomly with no internal compartmentation. To make it clearer, the bacterial cells, which of course are still present and abundant today, are all prokaryotic cells. Now, between 1.5 and 2 billion years ago, evolution takes off again, and in grand style, because a more complex type of cells appeared, the so-called eukaryotic cells. And these eukaryotic cells have an internal compartmentation, notably a nucleus, as everybody knows, and this is the case of all the cells of higher organisms. They have a set of organelles enclosed in membranes, the best known of which is probably the mitochondrion. The mitochondrion is an organelle capable of carrying out oxidative metabolism, which promotes the degradation of all compounds occurring in foodstuffs, making them react with oxygen. This turning point, namely the formation of eukaryotic organisms, paved the way to the appearance of multicellular organisms. As a matter of fact, virtually all multicellular organisms are eukaryotic. So the formation of eukaryotic cells is a prerequisite for multicellularity. And of course, the question is, how did the eukaryotic cells arise? In this case too, we know some details of the mechanism, but we cannot state we really know why and how they were formed. What we can say in simple terms is that eukaryotic cells resulted from the merging of various cellular components. In this respect, mitochondria are worthwhile to be mentioned again. Indeed, plenty of evidence supports the idea that they are remnants of ancient bacteria capable of sustaining oxidative metabolism. This process, the inclusion of a cell in another cell, is called endosymbiosis. Thus, the formation of eukaryotic cells also led to the appearance of oxidative metabolism in higher organisms, namely their ability to degrade carbon compounds, those our foodstuffs consist of, by organisms by reacting them with oxygen. This is an innovation of paramount importance in that it entails an energy release at least 10 times higher than that made available by the degradation of the same substances in the absence of oxygen, so within an anaerobic context. Well, the appearance of oxidative metabolism was undoubtedly triggered by a physical change in the atmosphere, namely the appearance of oxygen. Oxygen did not exist in the atmosphere until around 1.5 billion years ago. Its appearance is almost entirely accounted for by the metabolic activity of photosynthetic organisms, the precursors of today's plants, which are able to fix carbon dioxide and release oxygen. The appearance of photosynthetic organisms and of oxygen-releasing photosynthesis was the prerequisite for oxidative metabolism to occur. This may also be linked in some way to the appearance of eukaryotic organisms. even though the relationship between the two phenomena is not quite clear yet. This scenario highlights a key aspect for evolutionary dynamics, namely that the interaction and cooperation between large groupings of organisms have greater effects on the evolution of biosphere than those produced by a simple mechanism of Darwinian evolution. The appearance of oxidative metabolism completely changed not only the type, the diversity, the complexity of living organisms, but even substantially, it changed the physical aspect of the planet. This shows, therefore, that evolution is not merely a selection of the fittest. There are additional factors that I would say are even more decisive and unpredictable. I would like to comment a picture emerging from this story, stating that we can consider evolution as a parallel pathway of many organisms that change and modify independently from each other. And this is really decisive in the way they interact. And I really like this because we cannot really and properly see the evolution as a parallel pathway of many organisms that change independently from each other. What is really crucial is, with no doubt, how organisms react with each other. This story then continues with the appearance of the first multicellular organisms around 1 billion years ago. And this is another significant leap, as everybody can easily understand, even though at the beginning such organisms were basic. So they remained until around 500 million years ago, when another far-reaching event occurred, which wasn't precedented, nor did it occur again in the history of life. Around 500 million years ago, an era referred to as Cambrian Period by geologists, starting from the few existing multicellular organisms, all the large groupings of animal organisms rose. In zoological terms, they are called phyla. This included all the species that still live today, but also some species that later went extinct. Therefore, in a period that was quite shorter in the evolutionary time scale, there was a rather understanding and outstanding proliferation of life forms, so vast that it was dubbed as Cambrian Explosion. So the question is, why did such a wide and relatively sudden diversification occur? Again, no certain answers exist. Many theories have been proposed, including a combination of several factors, such as environmental ones, such as a glaciation, which actually occurred in that period, or variations in oxygen content, which substantially increased in that period. Or maybe, again, the appearance of ozone in the atmosphere, itself a result of the mentioned oxygen increase. Yet there may have been internal causes linked in some way to a trend of organisms to take the path towards greater complexity. What is for sure is that we have absolutely no certainties. It seems evident to me that, in any case, This journey highlights the complexity and unpredictability of life's paths. Besides the Darwinian selection, seemingly life did not evolve only through fighting but also through cooperation, as seen in the example of the eukaryotic cell in oxidative metabolism. In addition to that, as it is evident that environmental factors also played a role, are there other important factors as well? Absolutely, I would say. Based on our knowledge, both past and recent, in short, we can talk about another factor, which is substantially an intrinsic tendency of organisms to evolve. Researchers dealing with this topic have coined an English term that is evolvability, which is not easily translatable in other languages. It means the following. Organisms are designed to undergo diversification. If proper conditions occur, of course, it is a matter of an intrinsic trend rather than one linked to environmental components. But evidently, these two aspects coexist and set the direction of evolution. As for the tendency to evolve, one of the most remarkable examples in the case of OX genes is the following. OX genes produce proteins which in turn activate other genes, and these in turn produce other proteins controlling the development of body segments. Now, the outstanding feature of these genes, called OX genes, is that they are shared by a wide variety of species. These species are very distant from one another in evolutionary terms. And these OX genes and these species include the fruit fly, Drosophila, and vertebrates, including mammals and humans. Now, in the next footage, we're going to see how OX genes were discovered by Professor Lewis of the California Institute of Technology. He was the first to demonstrate the existence of such genes, while Professor McGuinness from Basel University demonstrated that these genes are present in humans as well as in the fruit fly. In 1995, the Nobel Prize for Physiology or Medicine was awarded to Dr. Edward Lewis and his collaborators for predicting the existence of this upstream regulatory gene, which was indeed identified later. To study this gene, Dr. Lewis decided to study the fruit fly as a model animal. Many researchers had already been conducted on this animal at the genetic level. First of all, Dr. Lewis began collecting the natural mutants of this fly. This one, for instance, has a pair of legs more than the normal fly. This one has no wings. This one has an extra pair of wings compared with normal flies. A particular technique was used to visualize the result of the gene's activity during the development of a normal fruit fly embryo. We can see that the larva develops in 14 separate segments, located one after the other. When the larva develops into an adult fly, the front legs form from the first thoracic segment, the median legs and the wings from the second segment, and then the back legs from the third segment. Thanks to these studies, Dr. Lewis was able to conclude that out of more than 10,000 genes, at least 8 determine which segments of the body will develop into legs or wings. The first homeotic gene identified by McGuinness was the one enabling the development of the second thoracic segment. This gene contained a key with a code of 180 letters. This key allows the gene to send messages to other master key genes, which lead to the development of the legs and the wings. In this fly, with 4 wings, preserved by Dr. Lewis, a mutation led the gene responsible for the second segment to extend its effects to the third, thereby causing the development of a second pair of wings. Dr. McGuinness then started to extend his research to other organisms, looking for that same code of 108 letters that would identify a homeotic gene. It was then that he made a great discovery. I remember that moment as if it were today. I was taking the X-ray film out of the bath, and I saw these black bands of human DNA. 8 homeotic genes were found in the fruit fly, but it was clear that roughly the same genes were also present in humans. In human beings, there are 13 different homeotic genes classified in 4 groups: A, B, C, and D. The numerous possible combinations among these groups produced a far more complex structure of the human body. When one of our hands has to get differentiated, first of all, the homeotic gene sends a signal to the master key gene responsible for the hand formation. In response to this signal, the cell division begins and the limb bud starts to develop. Then, at the hand level, a new signal is sent, ordering the self-elimination of certain cells. In this way, fingers get separated. First, the limb protrusion is generated, followed by the separation of fingers. This is how the complex structure of a human hand takes shape. At this point inside the limb, the homeotic genes responsible for the bone formation are also at work. The homeotic genes 9 coordinate the production of the scapula and the clavicle. The genes 10 coordinate the formation of the humerus, while the genes 11 start the differentiation of the ulna and the radius. The finger bones are formed under the guidance of homeotic genes A13 and D13 and grow as long bones. The wrist bone receives orders from gene A13 only and develops as a short bone. In Rose's case, it is believed that a mutation of gene D13 has prevented the complete development of a phalanx. The human body starts developing from a single fertilized egg cell. Both parents' genes are duplicated during each cell division so that all the cells of the body contain the same genetic program. Soon, the 13 different types of homeotic genes start to work to divide the embryo into sections. In each section of the embryo, several master key genes are activated and put in charge of producing, respectively, the hands, the heart, and all the other components of the human body. The master key genes activate signals to the gene they control to produce the proteins needed to form the various organs. As in a domino, the homeotic genes activate the genes downstream of the chain, and ultimately proteins are produced that will create the complex structure of the human body. As you have seen, the peculiar aspect of Huck's genes is that they are shared by organisms that are very distant from an evolutionary point of view. And what is even more surprising, although not explicitly mentioned in the footage, is that these genes developed 2 billion years ago, so long before flies and vertebrates appeared on the Earth. Thus, there is an evolutionary drive that generated these genetic models able to control and distract the formation and the growth of body segments of a wide range of species that only later would appear. And this is a very surprising exemplification of how in evolution, hence in organisms, evolutionary dynamics are embedded that go far beyond the Darwinian selection and that cannot be traced back simply to environmental influences. The latter play an obvious role but cannot justify or can do so just in a limited way the generation of diversity and wealth of forms we see today and of those we know have existed on our planet in the past. So there are clearly many factors playing a key role. However, only in a limited form they can justify the wealth and diversity of forms that we see today. So, considering all these aspects, do we have any idea on how the biosphere will evolve? Will evolution stop sooner or later? Well, I would like to first remark that the timing of evolution is far lengthier than the life of an individual. Generally, at least 100,000 years are needed for a visible change to be detected. However, two statements can be made with absolute confidence. Evolution is reversible and endless at the same time. It is reversible, as we see, for instance, in the case of the Cambrian Explosion. Many phyla that were formed have also disappeared and will never appear again. But it is also true that, as we know and constantly observe, the living species display an outstanding ability to adapt to new conditions. So, following the environmental changes, new species will also be formed to cope with new emerging challenges. I think there is no better comment to conclude than the sentence Darwin wrote whilst pondering over the beauty he perceived through his passion for the biological world. There is grandeur in this view of life, with its several powers, having been originally breathed by the Creator into few forms or into one. And that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning endless forms the most beautiful and most wonderful have been and are being evolved.